I'll just start off by discussing the linkage between heart failure and Alzheimer's disease. So cardiovascular risk factors such as heart failure, diabetes, and hypertension are strongly associated with the development of Alzheimer's disease, or AD. Vascular dysfunction has also emerged to be a major player in both the progression and development of Alzheimer's disease. And while precise mechanisms underlying these associations have yet to be fully understood, this presents a unique opportunity to develop models to enable the study of vasculature in neurodegenerative disease. So taking a closer look at the cerebral vasculature in AD, the brain is protected by a physiological barrier known as the blood-brain barrier. And clinically, the blood-brain barrier in Alzheimer's disease has been demonstrated to have several impairments, including reduced expression and impaired function of efflux transporters involved in the removal of potentially harmful xenobiotics, increased microglial activation and pro-inflammatory cytokine secretion, increased vascular permeability, and enhanced leukocyte transmigration, promoting a pro-inflammatory microenvironment. Further, patients with Alzheimer's disease have been shown to exhibit reduced cerebral blood flow or hypoperfusion, which can have detrimental effects to the blood-brain barrier. More specifically, shear stress from blood flow has been demonstrated to play important roles in maintaining the healthy physiology of the BBB, including enhancing barrier resistance, upregulating junction and transport genes, inhibiting cell proliferation, and facilitating reorganization of the cytoskeletal architecture. So taken together, this demonstrates the critical role that shear stress plays in capturing and modeling the cerebral vasculature. And this takes me to my project aim, which is to address the lack of in vitro dynamic models of the blood-brain barrier in Alzheimer's disease, which mimic the dynamic shear conditions and patient-specific characteristics. So in order to address this, the Simmons lab has developed the VitroFlow microfluidic platform for barrier modeling. So this is a well plate which delivers shear stress in a gravity-driven manner without the need for external pumps. So briefly, each of these units contains two adjacent channels in which barrier-forming cells, such as endothelial cells, can be cultured on the top of a permeable membrane, and on the other side, secondary or tertiary cell types, such as neurons or astrocytes, can be cultured. Once the plate is placed on a rocker, we can deliver unidirectional shear stress to the top layer of cells through gravity-driven transfer of media between the two reservoirs. And this platform in our research group has been previously applied to model the healthy endothelium as well as gut epithelium. So to extend on these findings, we were interested in investigating modeling the cerebral vasculature in a disease context. So in order to do this, we obtained stem cells from the New York Stem Cell Foundation from a patient with familial Alzheimer's disease and a corresponding control line, which was age and sex matched from a member of the same family as the patient with AD without the disease. We then differentiated them into brain endothelial-like cells and exposed them to physiological shear stress or static conditions and performed functional assays to assess their barrier function. So one of the first functions that we were interested in investigating was efflux transport function, as this is a critical mechanism in the line of defense against potentially harmful xenobiotics and toxins. So in the schematic on the left, after shear or static conditioning, we evaluated the efflux transport of three transporters: P-glycoprotein, breast cancer-resistant protein, and multi-drug-resistant protein, using receptor inhibition assays, in which barriers were pre-treated with a small molecule inhibitor for one of the three transporters, followed by treatment with their respective substrate. So what's reported on the graphs is fold uptake of the substrates compared to groups which did not receive the inhibitor, i.e., if transport was functional, we should see an increase in substrate uptake. And we can observe that under the static conditions, there is a significant decrease in efflux transport activity for the ADBCs, or the Alzheimer's disease brain endothelial cells, as compared to the control groups. And once shear stress is applied, these differences are no longer apparent, suggesting that the lack of shear stress reveals impaired efflux transport function in ADBCs. As one of the important features of the BBB is also to regulate the trafficking of immune cells, we next investigated the degree of monocyte adhesion in our barrier models. So under static conditions, we observed a significant increase in monocyte adhesion by ADBCs as compared to controls. And when shear is applied, there's a significant decrease in monocyte adhesion for all groups. This, again, suggesting that the lack of shear flow may elicit higher immune cell attachment in ADBCs as compared to controls. So finally, we also wanted to conduct proteomic assessment of the BECs and the F controls and the AD groups in co-culture with neurons. And so to do this, we differentiated BECs and neurons from iPSCs. We cultured them in either shear or static conditions for 72 hours before collecting the samples for mass spec analysis. And so while these results are still preliminary and analysis is ongoing, I wanted to share one of the comparisons that were particularly interesting and relevant in comparison to some of the differences that we were seeing in our functional assays. And so for these assays, we looked at collecting the proteomes from BECs in co-culture with neurons in static and shear conditions. And we found significant differential protein expression in the static groups as compared to shear. When we conduct pathway analysis on some of these differentially expressed proteins, we observe not only differential expression in pathways related to key endothelial functions like actin remodeling and extracellular matrix proteins, but also pathways related to cardiac diseases. And this may suggest that differential protein expression observed in our static co-cultures may share characteristics with that of vascular dysfunction and cardiovascular disease. So to conclude, the VitroFlow platform can be used to model brain endothelial barriers and demonstrate functional changes to the BBB. We demonstrate that shear stress may play a protective role in maintaining both efflux transport function and controlling leukocyte adhesion, whereas static conditions appear to reveal such alterations in ADBCs. Finally, this offers an avenue to conduct mechanistic investigations on vascular dysfunction in a physiologically relevant AD model with potential applications towards therapeutic discovery. And with that, I'd like to thank my research group, Professor Craig Simmons, for the mentorship and support, and all of our funding sources.